The thymus has extremely sophisticated functions, but only a simple structure visible on routine histological sections. So you'll be able to distinguish only two types of tissue, cortical and medullary tissue, and two classes of cells, thymocytes, T cells or T lymphocytes, and epithelial reticular cells. Note that from viewing this image that the thymus is divided into lobules, each with a darker staining cortex and a lighter staining medulla. These seams of connective tissue separate the lobules of the thymus and invest them with thin capsules. Look here at a cortical region of a lobule. The cells are predominantly T lymphocytes and their precursors. T lymphoblasts actively divide here and differentiate into small T lymphocytes. Most of the newly formed T cells will die at the hands of macrophages. Others, with appropriately rearranged T cell receptor genes, escape to the capillaries at the boundary with the medulla and contribute to the circulating pool of T cells. The only blood vessels in the cortex are capillaries, just identifiable in some parts due to the red blood cells in their lumens. This turns out to be very significant because there is a blood-thymus barrier in the cortex that is not apparent from light microscopy. See if you can see sparse supporting cells with larger pale nuclei. From their general appearance, they've been called reticular cells, but this is a mistake. They're actually epithelial in form. Their embryonic origins trace back to ectodermal epithelium of the embryonic foregut, and they retain their fundamentally epithelial commitment. Immunologists now subdivide these epithelial reticular cells into a number of different types, but they're all indistinguishable from each other in sections of tissue. There are also a substantial number of macrophages in the cortex, but you'll not be able to distinguish them from the epithelial reticular cells in this hematoxylin and eosin preparation. If a large pale nucleus has a substantial amount of cytoplasm around it, then it probably belongs to a macrophage instead of an epithelial reticular cell. So maybe here. The medulla is paler because it has far fewer thymocytes. This makes the epithelial reticular cells easier to observe. Some of them aggregate into easily seen pink structures called thymic or Hassel's corpuscles. The function and significance of these structures are unknown. Likely they have none. They are composed of swirls of epithelial reticular cells around a keratinized or a calcified core. The blood-thymus barrier is interrupted in the medulla, and here one can find blood vessels larger than capillaries. They then allow T lymphocytes to escape from the medulla into the general circulation. The thymus is largest during childhood and progressively involutes after puberty. This section is from an older individual. Most of the parenchyma has been replaced with adipose tissue, although some lymphopoiesis still continues even into the old age. Many large Hassel corpuscles are present, and these structures can continue to grow until some are even big enough to see with the unaided eye.